Hey Bio30s, I'm back again for another lesson. Actually, 1 second, I'll just turn on the lights here. Lighten things up a little bit, and we are going to start a new unit here. So we're going to talk about the male reproductive system, and we're going to discuss the structures of the male reproductive system, as well as the process of producing sperm, also known as spermatogenesis. So here we have the male reproductive system. So in this diagram, if you notice, there are a few parts here that aren't technically part of the male system. So for example, the urinary bladder, the ureters, those are part of the urinary system. They are separate, they have no reproductive function, but the urethra, which is part of the urinary system, is also part of the reproductive system in males. So that is why those two structures are included there. So if you look at the bottom, we start off with the testes. If you were to look inside of a testes, you would see these twisted tubules known as the seminiferous tubules. This is where the sperm would be produced. Right above the seminiferous tubules, we have the area called the epididymis. Also more twisted tubes. This would be where the sperm matures. This is where they would kind of mature, be fully formed, ready to go. Leading from each testes into the ejaculatory duct, we have the vas deferens, which we'll talk a little bit later about what a vasectomy is. It's actually just simply trimming or cutting the vas deferens. Then as we pass through the ejaculatory duct, we're going to pass by 3 glands, the seminal vesicle, the Cowper's gland, as well as the prostate gland. They're going to produce fluids that will make up semen. And then the semen with the sperm in it will then exit through the urethra. So when we look at the structures of the male system, basically at about the last 2 months or so of fetal development, the testes will descend into a pouch known as the scrotum, because sperm actually prefer about 2 or 3 degrees cooler than body temperature. So actually, if you want to have good sperm production, you have to make sure that the sperm are at a proper temperature, which once again is about 2 or 3 degrees cooler than the body. Generally in nature, it's not a great idea to have your reproductive structures on the outside of your body, easy to be damaged, right? But in this case, it is important because if they were inside of the body, they would be way too warm, and actually you wouldn't be able to produce sperm properly. There is one of the things they look at too is if you go to, say, a fertility doctor, say you're having difficulty with your fertility, one of the things they'll look at is actually what type of underwear you wear. They'll actually check to see, hey, are you wearing underwear that might be too tight? Is it too warm down there? Because that could be impacting your fertility. It is actually something they look for there. So sperm, they need a nice cool temperature, not too cold though. If you've ever jumped into a cold lake before, you know what I'm saying. But we want it just 2 to 3 degrees cooler, and that means you have good, healthy sperm being produced. So from the testes, we then have the vas deferens, which will then lead into the ejaculatory duct, which once I said, like I said, which will push sperm and the fluids known as semen into your urethra. So let's talk a little bit how you make sperm. So the process of making sperm is known as spermatogenesis. So spermatogenesis involves the creation or maturation of sperm. To do that, you're going to need some nutrients, you're going to need some oxygen, you're going to need some hormones. Well, here's the thing. Where are those things going to come from? It's going to come from your blood, it's going to come from your body. The thing is, though, you don't really want your blood and your sperm mixing, because actually if your sperm were to get into your bloodstream, you might make antibodies and your immune system might attack it, which obviously you don't want. So you have these cells called Sertoli cells. They will actually surround the developing sperm cells, and they act as a barrier between your blood and your sperm. So the Sertoli cells will allow nutrients, hormones, oxygen to diffuse across them into the seminiferous tubules where the sperm are being produced. So very important we have that barrier. Now we start off with a cell known as a spermatogonia. So these are the sperm producing cells that line your testes. These spermatogonia can divide through a process called mitosis, which we'll talk about a little bit later. That's just simple cell division. So 1 spermatogonia can make 2 spermatogonia, 2 can make 4, 4 can make 8, so on and so forth. Now, what the spermatogonia can also do is it can divide into a primary spermatocyte, which would still have a full set of DNA, would still have a total of 46 human chromosomes. But what that spermatocyte can do is it can then undergo a process called meiosis. During the process of meiosis, which we will discuss in future videos, during the process of meiosis, you're going to take that primary spermatocyte, you're going to split it not once, but twice into 4 cells. And these 4 cells are going to have half the number of chromosomes. So it's going to look a little something like this. We start off with a spermatogonia, which would be these cells right here. They would divide into a primary spermatocyte, which once again would still have your full complement of chromosomes. A primary spermatocyte would divide into secondary spermatocytes. This would be when we half the number of chromosomes, 23 into one cell, 23 into the other. Then those spermatocytes would divide into spermatids. Once again, another division. So we end up with 1, 2, 3, 4 cells, each with 23 chromosomes. And those spermatids will eventually become sperm. Once again, 23 chromosomes apiece. When you look at a sperm cell, it's really a simple cell, honestly. It's a head. Inside of the head, you'll notice there is a nucleus. So that's where the DNA is stored. And there's also a little cap with enzymes in it. So that's called the acrosome. That will actually help the sperm burrow and enter the egg if it were to come in contact with an egg. There's then the midpiece, which is full of mitochondria to produce ATP. And that ATP is then used to power its tail. So it's a pretty simple cell, honestly. Its job is very simple, to deliver those 23 chromosomes into an egg. To get the sperm to the egg, it does need some help. So it's going to need some fluids and some protection. So semen would coat the sperm, kind of like armor, if you want to think of it like that. So there's 3 glands that produce semen. There's the seminal vesicle, there's the prostate gland, and there's the Cowper's gland. So the first gland we pass by is the seminal vesicle. So the seminal vesicle will produce fructose and prostaglandins. Now fructose, you can probably guess what that's used for. That's essentially sugar. So that's going to power those mitochondria, that mid-piece, to move our tail. Prostaglandins, those are used to actually cause muscular contractions to pull the sperm into the female reproductive system. Interestingly enough, the female will also make prostaglandins during childbirth, and this would be in the opposite direction to push the baby out the reproductive system. Then we have the prostate gland, which makes an alkali buffer. The vagina is a low pH, so an acidic environment, not very good for sperm. It's pretty hostile for sperm. So they have that alkali buffer to protect them against that acidic environment. And then finally we have the Cowper's gland, which is going to produce a mucus, which is going to coat the sperm, further protect it, allow it to swim, and allow it to have a little bit of mobility. So those 4 fluids, along with sperm, would make up semen. And then sperm, if properly produced, so if it was, and at the end of the day, you make tons of sperm, like millions upon millions of sperm. Out of the millions upon millions of sperm, like let's say in one ejaculation of sperm, you produce anywhere from 200 to 400 million sperm. Like that's a lot of sperm. Out of those 200 to 400 million sperm, you're lucky if 15 to 20% of them actually are viable to make the egg. It's really a quantity versus quality. It's a lot of sperm, but only a few of them are actually like viable to end up getting to the egg. Now, the viable ones, they can live anywhere from 24 to 72 hours as they make their way to the egg. That being said, if you were to freeze them, they can last for many, many years. You can actually freeze your sperm and or thaw them and actually use them years later. So here's your basic pathway. You have your seminiferous tubules, your epididymis, right? That's where the sperm mature. Seminiferous tubules are where they're made. We have the vas deferens, that's where they're transported. They are going to pass by the seminal vesicle, where they're going to pick up some fructose and prostaglandins. They're going to pass by the prostate gland and Cowper's gland, where they're going to get an alkali buffer and some mucus, and then out the urethra. So next lesson, we are going to talk about the male hormones, so where the male hormones come from, and what each hormone does, and then how it's related to spermatogenesis. So thanks again for watching, and we'll chat soon.